So this is another way for you to look at it and an easier way for you to study because it gives you some ideas of what's going to happen. If you look at this, we've got our receptor, and then we're going to get cyclic AMP, cyclic GMP, we're going to get calcium, we're going to get diacylglyceride, or we're going to get a protein kinase, which is a separate kind. Okay? So there's your possibilities of what can happen when the alpha subunit separates. Okay? Those are all the possibilities that can happen, and this is the kind of thing you want to study from. Okay? You may not have to know every single one of those little proteins along the way that the ball gets passed to, but you want to know what your output is going to be at the end. Okay? You want to know where the messengers are that are actually going to be doing the work. Okay? Which what we will call, what we're going to call the, come on, there we go. What we're going to call our effectors. Okay? Okay. Okay, so this is a specific kind of a G protein couple receptor, and it applies to you guys on a daily basis, so I'm going to explain some of the things that are happening here. If you are stressed, now, in the olden days, back to the cavemen, the stress would have been fight or flight, right? They're being attacked by a mammoth or something, okay? Fight or flight. So your body thinks it's got to get ready to run, or it's got to get ready to attack, okay? Fight or flight. Nowadays, we may be really stressed, which could translate into waking up at night and thinking about all the things you've got to do. Or maybe you're really stressed because you can't possibly get all the stuff done in a day, or because your friends really want to go to the movies and you're supposed to be doing your homework. That's stress, okay? But your body doesn't know the difference, and your body still acts like being under stress means that you better be ready to go, okay? So epinephrine is one of the neurotransmitters, sometimes also norepinephrine. One comes from your adrenal gland, one comes from your brain, and that is going to be saying it's time to make some energy, okay? So in this case, it's going to bind to the transmembrane, and it's going to activate its G protein right there. And that G protein is going to move over, the alpha subunit will move over to adenylate cyclase, and it's going to make cyclic AMP. And the cyclic AMP is going to then be the effector that goes forward so that it can increase your heart rate, dilate your skeletal muscles so that they have more blood, so that they're ready, they got oxygen, and they're ready to pump and run hard. And it's also going to, and this is really important, it's going to break down the glycogen, which is stored glucose that's in your liver, because it knows you're going to need more energy. So it's going to do that. Well, now what if all your stress has nothing to do with physical? Something has stressed you out. Maybe you got a phone call from your home and your parents are mad at you. You're all stressed out. But you're not running around and you're not spending any physical energy, but you have now got all this extra sugar hanging around. The next thing you know, you're going to be exhausted. You're going to be so tired, you're going to think, oh, I've got to have a nap. Then you're going to think, I didn't do anything, but I'm exhausted. Okay? That's why. Because your body was under stress and it's behaving like that stress should have involved you running away or fighting a tiger. Okay? Okay. When I say cholera, raise your hand if you know what cholera is. Anybody know what cholera is? Okay. I ran into this last year, so I'm helping you, but don't laugh at me. Okay. And have you ever heard of Hija? So Hindi and Nepali is Hija. If you are from Thailand, it is Ahi Wakaru. If you're Mali, it is Tawon. And that's all of them. That's all I wrote down. Okay, so cholera is a toxin that comes from a bacteria. It is very common for there to be cholera epidemics in locations where there's like flooded water or in areas where there's poor hygiene. Oftentimes, if there's a natural disaster and people have disrupted water supply, then you have the chance of getting cholera. Okay, now do people remember what it is? Do you know what cholera is now? Okay. Okay, so cholera is very dangerous because a lot of people die. Okay? And the reason they die is because although you can live through it, you have to get treatment for it. Okay? So, the bacteria comes into your body and it's in your intestine. Like I said, it comes in the water. Okay? The bacteria comes in the water. So it's not necessarily the bacteria that's going to make you sick. It is a toxin. Oops. It is a toxin that the bacteria is going to put out. And that toxin is small enough that it can actually go inside your body. It is going to find a receptor to bind to. Now, this is sneaky because that's not what the receptor is supposed to bind to, but it just happens to fit. Okay? So, it binds to that receptor and it gets internalized. See this? It says endocytosed. It's going to get carried into the cell. And then the toxin is going to go to the G protein and it's going to activate the G alpha stimulating subunit. Okay? Now, this is the tricky part. It's a toxin and it's stuck there and it's never turning off. Okay? There's no homeostasis. There's no balance. It's going to stay on forever. Okay? Until the cell dies. It's going to stay on forever. So now you are going to have a lot of cyclic AMP made. Way too much. 100 times more than you normally would have made. And that cyclic AMP is going to go over here to this membrane receptor, which is called CFTR, and it's going to make that active. So when this is active, it's going to let all kinds of chloride out of the cell. And that is going to make the sodium and the water leave the cell. Now this is in your intestine. Now you got all these cells in your intestine letting out all your nutrients. They're letting out your water, they're letting out your sodium, they're letting out your chloride, your potassium, and it's all coming out as runny diarrhea. Now, there's no way to stop this, okay? There's no way to make that toxin stop. Once it gets in there, it's going to keep going until the cell itself dies, okay? So, what happens is that people become completely dehydrated and then they die, okay? Then they have such an imbalance in their potassium and the calcium in their heart, makes the heart go crazy and they die. Okay? The way to keep that from happening is simply to replace fluids in their body until the bacteria dies. The bacteria will die. It's self-limiting. If you can keep the person well hydrated, if you can replace their nutrients like Gatorade or 10K or any of those sports drinks, that's the idea. They're going to replace your nutrients. That's what you're going to do. You're going to try and replace that so the person has a fighting chance to live. And eventually, the bacteria will die and there will be no more toxin made. Okay? But that all starts because of the fact that it's attached to a G alpha stimulatory and it doesn't stop. Okay? Okay. So now we have a little movie. Then after the movie, we'll have a little 10-minute break since we already heard I already heard the bell go. Okay, let's see if we can make this work.
Hi, it's Mr. Anderson, and welcome to Biology Essentials, video number 38. This is on signal transduction pathways. Signal transduction pathways are very important in the actions of cells, but they're sometimes misunderstood. So I wanted to start with an analogy. And so when Jimi Hendrix plays the guitar, he would vibrate the strings on the guitar. those would then be transduced. In other words, the pickup in a guitar, the electric pickup in a guitar, has magnets and wires inside it, and what it does is it transduces that message of the wires into an electrical signal. It then goes into an amplifier where you can make it really, really loud, and so we can hear it, and that's what he was famous for. And so signal transduction pathways in cells do the same thing. It starts with a message, and that message is in the form of a chemical message, and then that's transduced into actions within the cell, and it also can be amplified. And so signal transduction pathways all work in the same way, and there are a couple of ways that their actions work. Sometimes they'll actually modify a protein or change the shape or the conformation of a protein, but mostly what they'll do is there'll be what's called a phosphorylation cascade. In other words, a phosphate group, which remember carries energy, will be passed off from one chemical to another to another to another until it eventually has an action. And protein kinases are important in that. And so the example that I'm going to give you today, a message comes in, a message is picked up by a receptor, and so this line we could say is the cell membrane on the outside of the cell. That message will dock with the receptor, and when it does that, in this case, it'll change the shape of that receptor. The example I'll give you is what's called the G protein receptor. Then we'll have transduction. Transduction, remember, is when we're switching that message and we're changing that from a signal message on the outside of the cell to a message within the cell. We use what are called secondary messengers. The one that I'll give you an example of is called cyclic AMP. It's a very common messenger in cells. And then that'll eventually target the cells. In this case, it's going to target cells in the liver, and it's going to make them release glucose from glycogen. And so that's kind of a signal transduction pathway. It starts with a message, and then it eventually has some kind of a target within the cell. And so let's get started. This is an animation of this typical liver cell and how epinephrine can affect it. And so we're going to go through, and I'll pause at a couple of different spots and explain it. So epinephrine is the messenger. Epinephrine is going to be given off from the adrenal gland. It's going to move throughout your body, but it's going to especially affect cells in the liver. So epinephrine is going to dock with this receptor, and in this case it's called a G protein receptor. And so the epinephrine is what's called a ligand, and so a ligand is going to be a chemical. It's a chemical that can't make its way through this cell membrane. It can't make it through this hydrophobic region, so it's going to dock with the G protein on the outside. G protein is a protein that's embedded within, it's a snaky looking kind of a protein. It's embedded within that cell membrane. So it's got a portion on the top and it's got a portion on the bottom. It's got these units on the bottom. They're called subunits. And so what happens is when that ligand attaches, in this case epinephrine, with the G protein, it causes a conformational change in that protein. So it's changing the shape of the protein. And what happens when it changes that shape is it actually releases one of those alpha subunits. And so one of the subunits, called the alpha subunit, will be released, and it's going to move to a protein just right down the way in the cell membrane. This protein is called adenylyl cyclase, and it'll make sense why it's called cyclase in just a second. But essentially what it is before the actual alpha subunit comes, it's an inactivated enzyme. In other words, it's an enzyme that's not working yet. It hasn't changed its shape, so it's actually a functioning enzyme. But once the alpha subunit is in place, it's ready to do its job as an enzyme. And in this case, what it does is it converts ATP, adenosine triphosphate, into cyclic AMP, or we sometimes call this CAMP. Now, what is ATP? We know that has 3 phosphates attached on the outside. It carries energy mostly in cells. We're very familiar with how ATP can be converted to ADP when it drops off one of those phosphates, but it can also drop off 2 phosphates, and that's what happens in this case. So now it becomes AMP, or monophosphate, but there's also a cyclic portion to it, and so it adds right where we come off of the sugar. It's actually going to make a cyclic portion, and I'll put a picture in here of that molecule, ATP, and now we've created these messengers. Messengers are going to spread throughout the cell, and this is called cyclic AMP, and those secondary messengers in this case are going to target something called the protein kinase. Protein kinase, it's made up of a number of different subunits of protein, but kinase means it does something, or it does action. In this case, this protein kinase is going to have 2 catalytic subunits. Catalytic catalytic means things that are going to speed or speed up chemical reactions. And then it has these two regulatory subunits. And so as long as the regulatory portions are attached to the catalytic portions, protein kinase is inactivated. It's not going to do anything. But let's watch what happens to the cyclic AMP. It'll actually bind to those regulatory portions of the protein kinase, and it releases the catalytic portions. And so now we have this cascade. In other words, we have this cascade of energy. Those catalytic portions are going to become phosphorylated. In other words, they're going to pick up energy from ATP, and they're going to become activated. And they change now from a green to kind of a yellow activated color. They then can act on enzymes within the cell. Sometimes they'll act through a number of different molecules within the cell. In this case, it's going to drop off that phosphate to phosphorylase, and it's going to activate phosphorylase so it can release glucose from glycogen within the cell. Now, once we don't have that ligand attached anymore, we don't make that cyclic AMP, then the whole thing is going to shut down again. And so the signal transduction pathway is simply a way that we can take this message and we can move it throughout the cell and then have desired consequences within the cell. Okay, so let's do a little review. And if you've ever watched the show Dora the Explorer, there will be times where she just kind of pauses and looks awkwardly at the person who's watching the show. And so that's where you have to jump in and help a little bit. So let's do a little bit of review. So we start with this at the top. We've got epinephrine at the top. And so what do we call this? That's right. It's called the ligand. A ligand is a chemical that can't make entry into the cell, but it's going to attach to the receptor. So this is called the receptor. Do you remember what that's called? That's right. It's called the G protein. And so what will happen is that ligand will attach to the G protein. It's got a number of different subunits. This one right here on the end is called the alpha subunit. That's right. Hey, if you're not getting any of these, you may want to go back and watch the earlier portions of the video. We've got this over here, and this is probably the one that you're going to struggle with the most. What's the name of that? Oh, that's right, adenylyl cyclase. And so what happens is the alpha subunit is going to attach to adenylyl cyclase. We then have an enzyme that's functioning. It's going to take in these little starbursts. Those aren't starbursts. They're called, that's right, ATP. ATP will then be converted to cyclic AMP or CAMP. And cyclic AMP are now going to go work on this protein kinase. That's right, protein kinase. It has two portions that are catalytic and two that are regulatory. That's right. Okay, so the cyclic AMP is going to move over to that. But let's watch what happens for a second. Because there's not just a few cyclic AMPs in a cell. There are going to be lots of cyclic AMPs and lots of protein kinases in the cell. And so remember when we talked about our analogy of Jimi Hendrix, this is where we can amplify the message. So we just have this one ligand up here, but we can have all this action going on. I didn't want to animate all of that, so I went back to just one protein kinase. So we will free up the catalytic portions. We now add energy to them. What's that called? Phosphorylation. That's right. We're going to add a phosphate group to them. They're going to turn yellow. And now they're able to pass on that phosphate group to phosphorylase. So it's activated, and it can break down glycogen into glucose within the cell. And so that's the signal transduction pathway. It's fairly simple. It's got a lot of steps in it, but it's just like playing the electric guitar. And so I hope that's helpful.
All right. Okay, so now if you have to draw that whole thing on a test, you'll be in good shape, right? You got it? Okay. Your name and your roll number needs to be on the top. There's 3 questions, so just put your name and roll number on the top and then do 1, 2, 3. Okay, so here's question number 1. Okay, we're ready for question number 2. Okay, we're ready for question number 3. All right, so we'll pick up the cards and then we'll see what the answers are. Okay? And after you give up your card, you can talk to your friends. Oops. Would you get that for me? There, you got yours? Okay. You got everybody? You got an extra? Okay. Question number 1. Who thinks the answer is A? Everybody thinks the answer is C? It is. You did good. Okay. All those who said B? Who said B? Who said C? It's C. That's right. If it's hypertonic, that means it's going to be low water. Then the water is going to go out of the cell and into the outside. Okay? Oh, we got a few that did we get pick up? Okay, last one. Okay. So if you thought it was D, you've now been convinced that it's actually B, and B is correct. Okay? And that's the key. Okay. Okay, it's my turn to talk. So I hope that you have an understanding and now what the questions are going to be about. It's going to be about the stuff you just learned the day before, okay? And it's to make sure that you refresh that information before you walk into class so that you'll remember the answers. It's not killer hard, although I can tell this is a smart class, so I might have to up my game a little bit and see if I can come up with something that might make you think a little more. All right. We are going to talk about some other pathways. I know there's a lot of this. I know that means there's a lot of repetition. I'm hoping as we go through some of these that we can talk about some of the real life examples so that you'll get the idea of how these things work. This is the pattern of what happens to the alpha and gamma subunits, okay? Or it also is the same thing that happens to the alpha Q, okay? So alpha Q and the beta gamma can both do the same thing, and that is they're going to go and activate phospholipase C, which is the PLC, okay? Phospholipase 3, what breaks it into two pieces. The two pieces are PIP2 and DAG, which is diacylglyceride, okay? It's going to break into those two pieces. The DAG, the diacylglyceride, is going to go and activate protein kinase C. But the PIP2 actually changes into PIP3, and it's going to go let out calcium. Calcium is incredibly important as a second messenger. Calcium is critical for your heart, and calcium is critical for your muscles, okay? So most of the time, any calcium that's in your body is very tightly stored away. In this case, it's showing that it's stored inside the endoplasmic reticulum. When we talk about muscle cells, you'll remember that it's called the sarcoplasmic reticulum. So the storage, the calcium is stored in here because as soon as the calcium goes out, it's going to start a multitude of effects in the cell, okay? So that is how we're going to get the calcium out. We're going to get the calcium out with this pathway, okay? Okay, there's a little learning slide for you so that you can remember when you're trying to memorize this stuff. So that you've got your adenylate cyclase, which is the same as adenylene cyclase, and ATP are going to make the cyclic AMP. You've got the phospholipase C, which is going to turn into the IP3 and the diacylglyceride. You're going to have the guanylate cyclase, which is going to make the GTC, okay, the GTP. And the cyclic GMP. And then you've got the phosphodiesterase, which we haven't really talked about yet, but also makes the cyclic AMP, okay? All right? So that's your little slide for when you're trying to remember how the processes go. Okay? And then the last part, when you get to the effector, when you get to the actual kinase that's going to go and phosphorylate, we've got the PKA, There is actually a PKB, but it's not usually called PKB. There's PKC. Anytime you see that C, it probably has to do with calcium. Okay? PKC usually has to do with calcium. GMP, calcium calmodulin. Calmodulin is another effector. It's another protein that is activated by the calcium hooking to it. And then down here at the bottom, this NO stands for nitric oxide. Nitric oxide is one of the few actual gases in the body that is also a second messenger signaling system. Okay, so there are a number of other kinds of G proteins. One is a really small one that only has one unit, and that is the one that you see all the time called a RAS protein. If you think back to that very first slide, let's see. There you go. You see all the RAS proteins on there? Like I said, whoever finds these things gets to name them, and then after they named them, they found out that it was really similar to the alpha subunit, and so now they recognize that it has two points. Okay. And the other thing to know is that there is actually a channel. It's a potassium channel that is connected to the G protein in some cases of the G protein. And that means it is going to let potassium go out of the cell, okay? Or in this case, inward in the cell, depending on how much potassium is actually in the cell. Okay, so there's a channel for potassium to go through into the cell and hyperpolarize the cell. Next week, we're going to talk about what hyperpolarizing the cell means. So that'll be coming right up. Okay. Again, this is for you, for your understanding, so that when you're looking at this, you'll be able to understand the process of how things are going. And this is the links between the second messengers and the phosphorylation at the end. So you've got the G protein, you got the G effector, the second messenger, and the protein kinase at the end. It's just another way of presenting it so that you have whichever learning style works for you works. Okay. I mentioned the tyrosine kinase before, and now we're going to talk about it in more depth because of the fact that it has such a significance in your body. Okay. It is connected to most of the growth factors. It is connected to a lot of cell division. That means that when things go wrong, they can go really, really wrong, okay? So there's a list there of some of its ligands on the side, the epidermal growth factor, the nerve growth factor, platelet-derived growth factor, hepatocyte growth factor, fibroblast growth factor, and insulin. And we'll talk about the insulin a little bit in a minute. But you can see, looking at those things, when you're talking about a growth factor, you can imagine all the possibilities that can go wrong when you are talking about overgrowth. So, for example, the fibroblast growth factor, fibroblast is part of a scarring process. Sometimes people have scars and they just keep growing bigger and bigger and bigger, and they keep consuming more of the tissue that shouldn't have been a scar. It's like it shouldn't have been anywhere near that big. Usually the surgeon ends up cutting out the entire scar and trying to do a do-over, basically, so that they can hopefully get that pattern and get it to slow down.